Hello everyone, welcome to today's lecture on bacterial growth curve. In this session, we will explore the different phases of bacterial growth, how to measure growth, and analyze experimental data to understand microbial dynamics better. Today, we'll learn about bacterial growth curve and its four phases, measuring bacterial growth using optical density, and conducting an experiment to track bacterial growth trend over time. Bacterial population change over time in a controlled environment, following a predictable pattern known as the bacterial growth curve. This curve consists of four distinct phases: lag phase, exponential phase, stationary phase, and death phase. To measure bacterial growth, various methods can be used, such as optical density, dry cell weight, and colony forming units. Today, we will focus on OD600 as our primary measurement method. The lag phase is the initial stage where bacteria adjust to a new environment. There is no cell division during this phase. Instead, bacteria are metabolically active, increasing in size and synthesizing RNA, enzymes, and growth factors required for replication. This adaptation phase allows bacteria to prepare for exponential growth. Following the lag phase, bacteria enter the exponential phase, also called the log phase, where they divide rapidly. The bacterial population doubles at a constant rate. This phase continues as long as nutrients are available and waste accumulation is minimal. Bacteria are at their healthiest and most active state, making them ideal for use in experimental studies. When plotted on a graph, this phase forms a straight line, and the slope represents the specific growth rate of the organism. The stationary phase occurs when bacterial growth slows due to nutrient depletion and waste accumulation. At this stage, the growth rate equals the death rate, leading to a stable population. Toxic compounds build up, limiting further bacterial division. On a graph, this phase appears as a horizontal line, indicating that the population size remains constant. Eventually, bacteria enter the death phase, where the death rate exceeds the growth rate. Key causes of cell death include lack of nutrients, unfavorable physical conditions, accumulation of toxic byproducts. As a result, the bacterial population declines over time. To quantify bacterial growth, we use optical density OD600, a method that measures light absorption to estimate cell concentration. Why do we measure OD600? Bacteria absorb light most effectively at 600 nanometer, making it the optimal wavelength for measuring growth. OD600 is a non-destructive, real-time method allowing continuous monitoring of bacterial cultures. Now, let's look at the experimental setup for measuring bacterial growth. First, inoculate 100 microliter of bacterial culture into 10 milliliter TSP. Measure OD600 at the start of incubation. Incubate the culture in a shaking incubator at 37 degrees Celsius. Measure OD600 at 3, 6, 12, and 24 hours to track growth. Record the data and analyze bacterial growth trends based on OD600 values. After collecting OD600 values, We plot them on a graph to visualize the bacterial growth curve. The resulting curve should display the lag, exponential, stationary, and death phases. By analyzing the slope and shape of the curve, we can determine bacterial growth rates and the effect of environmental factors on the cell survival. In summary, today we explore the bacterial growth curve, its four distinct phases, and how OD600 measurement is used to quantify microbial growth. Thank you for your attention.